this is the 7th annual session. We started in 2008 at the World Stem Cell Summit in Madison, Wisconsin, where we have Escro, Scro, IRB discussions. And it's not something that we have thousands of people attend. It's usually a handful of people. But as organizers of the World Stem Cell Summit, we do this because it is a service to the community. And we want to have an opportunity in each region that we go to for those that are interested to be able to be involved with it. And Melinda Abelman has, of course, over the years been involved with every single one of these. And it's highbrow stuff. I'm a member of 2 escrow committees, one at the University of Miami Miller School of Medicine, and also now I've just signed on for VGTI's Florida Vaccine Gene Therapy Institute is now going to start doing stem cell research in affiliation with the Karolinska Institute in Sweden. And so I've signed on. I know the folks there, and Richard Jove, who has moved from City of Hope to be the head of science there and really build that program up. So I just want to thank all of you for attending this, and it's important for us as a profession to move forward, and I hope we can continue with this program. As long as we're doing World Stem Cell Summits, there's always going to be a space for this session. So I just want to thank you for attending, and Melinda, the floor is yours.
I'm happy to have Bernie take some of my thunder by commenting on the fact that this is the 7th annual time that Bernie has generously provided time in the program for this. I think that the fact that the oversight role at our research institutions has garnered some time at this meeting is a reflection of something that I would like to see change, and that is that the research is viewed as exceptional. I look forward to a time when the stem cell research is not being conducted against a backdrop of the politics that it has been for all of its duration. It is also, I think, a reflection of the fact that we do not have a national framework in which the research is conducted. It's the vacuum of not having national regulations. And we find that institutions are operating in silos, in some cases in a vacuum. So this meeting does provide an opportunity for people from different parts of the country to come together. I recognize most of the faces out there, so it's also a time for people to touch base about how things happened in the last year. Melinda and I, entitled today's session, Evolving Roles. That's a result of the fact that we are aware of the roles that have changed for our own escrows, and we were struck by the different ways that the escrow at our institutions have evolved, made us wonder if and how escrows and scros or whatever committee you have that's looking at this research has evolved, hoping to get information back from you. In the absence of a national guideline or regulations, it's even more important for us to be coming up with best practices and to benchmark against each other, or at least know what other institutions are doing to help inform the discussions and the decisions that are made at our own institutions. So we do hope that you will participate. It's a small group, and let us know how things are going at your institutions. Bring your information to us. We are. I think this is that we are looking for the feedback that comes from the varied expertise that's here in the attendees in the meeting today. This is how we advertised this session. We hope we'll deliver on it. They asked for an abstract. That's what we provided. And what we plan to do is to have an overview of the few regulations, the guidelines. Melinda is going to provide that. I think that that's going to be very familiar to a lot of people in the room, but it continues to be sufficiently germane to the conversation, and I think it's well worth the time to go back over the current situation with regard to regulations and guidelines and the history of how this all evolved. And then I'm going to talk about the escrow that serves Rockefeller University and, well, actually, oversight of stem cell research at Rockefeller, and use that as a backdrop to bring up a couple of topics that I hope to get your feedback on. And then Melinda's going to do the same thing, giving a case study from partners, and go over another couple of questions. Okay, Melinda.
Okay, I thought it would be a good idea to start off with a little bit of the framework of the oversight, because we are all sitting in our own institutions and we get used to following the rules that our own institutions do, and we don't always see the big picture of what's going on. So the framework are the federal laws, and which have to do with human subject protection, animal welfare, biosafety, and funding restrictions, the FDA. But basically, there isn't any overarching regulation or law for stem cell research. There are voluntary professional guidelines, the National Academies of Science, and the ISSCR, the International Society of Stem Cell Research, and you'll hear more about those, and state laws that vary by jurisdiction. And how do I do this? And where do I point it? Okay, thank you. So I like to start off with this because I like this slide very much. It reminds me of Sesame Street. Three of these things are kind of the same and one is different. A little game they used to play on Sesame Street, and they probably do. And you can see that the other oversight bodies, the biosafety, the human subject protection, and the IRB, and the animal welfare, all have well-established federal mandates and maybe state statutes and regulations that go with them as well. But as far as Escro and Scro, there is no federal mandate at all. And there's a possibility in your state some sort of law or regulation or a funding requirement. So I wanted to talk a little bit about the unstable nature of our federal regulatory framework. The 1966 Dickey-Wicker Amendment to the Health and Human Services Appropriations Bill is the backbone of our current embryonic stem cell policy. It states that you cannot harm or destroy an embryo for research purposes. But how to interpret this? Through the years, there's been a succession of executive orders and guidelines. In 2000, Clinton released some guidelines. Right after that, very soon, President Bush had a directive, and then Obama had an executive order, and they all interpreted this idea of harming and destroying an embryo in a different way. It's very political. In 2009, the Obama administration issued the NIH guidelines for stem cell research, which we'll talk about a little bit. And this is not stable either. There's a national registry of stem cell lines. There was a very contentious 3-year challenge, Shirley versus Sebelius, to the NIH guidelines and the federal funding of stem cell research. And finally, the Supreme Court allowed the guidelines to stand by not hearing the last appeal. So, so you could see that there's nothing very stable about our situation now, and it's very politicized. Just very briefly about the NIH guidelines. Escrow is not mentioned at all. It established a new NIH registry of HESC lines that are eligible for federal funding. Funding is to use embryonic stem cells that were derived from IVF embryos created for reproductive purposes in excess of clinical need. There's detailed consent requirements. There's a whole process for having the lines listed on the registry, and some of the uses are limited, and some are, you can use them freely. I mentioned the early Harvard University lines, which are basically limited to funding for diabetes research, because they are in widespread use at our institution. You have to consider state laws. There are permissive versus restrictive states, and there are states that are totally silent regarding stem cell research. The focus of the laws and regulations are on funding, reproductive and therapeutic cloning, informed consent requirements, compensation, donor compensation, and the type and the scope of the oversight. The International Alliance for Stem Cell Research is active with a group of the more permissive jurisdictions, and they did a comparative analysis of these state policies, and they showed the policies were similar, but there was enough variety to pose potential barriers and risks to collaboration. So there would be differences in whether a state will allow compensation to an oocyte donor, or maybe difference in what is required in consent forms. Okay, or SCNT, or there are differences. I just wanted to mention the IASCR one more time because there's interesting information on its website. We are, as Amy said, we're operating in silos with little communication, and there's actually on the website there a 2011 survey that tried to understand what escrows and scros are reviewing and how they're operating. So the National Academies of Science, the guidelines, were issued in 2005, and they're voluntary. And many institutions welcome these because they're such a patchwork of regulations, and they really welcome them, and establishing escrow committees. In 2008, the guidelines expanded to include other human pluripotent stem cells, including induced pluripotent stem cells. In 2010, the guidelines were amended to recognize the NIH guidelines that were instituted after President Obama was elected, and it also acknowledged the divergence between some of the recommendations of the NAS guidelines versus what the requirements are of the NIH. So there are some differences there that I don't have time to get into, but they're very interesting. And of note, 2010 was the final report by this committee. So there's no further central guidance by any national body in this field that's rapidly evolving. The original recommendation said there should be a national body that's going, and it does not exist. Just a few other things about these amendments. They added the use of non-embryonic pluripotent stem cells. And it did say, because non-embryo derived stem cells are derived from human material, their derivation may be covered by existing IRB regulations concerning review and informed consent, depending on the source of the tissue used. No escrow committee review is necessary, although the IRB may always seek the advice of an escrow committee if this seems desirable. Where appropriate, that's not my underline, that's in the guidelines, the IRB review should consider proper consent for the use of the derived HPS lines. Some of the recommendations for informed consent that apply to HES cells also apply to human pluripotent cells, and there are a number of things that, like commercial development, and there are a number of things that if you're banking these cells, you should really advise your investigators to consider putting in their consent forms. The other thing is that HPS research designed or expected to yield gametes should have escrow review, and some HPS chimeras require escrow oversight if the cells could integrate into the germline or brain. And again, these are all voluntary recommendations, and escrows aren't mandated anywhere. This is the International Society of Stem Cell Research in 2006. It's a broader scope than NAS. They actually did include human pluripotent stem cells. And I thought it was amazing when you read them. It's almost as though they already existed, induced pluripotent stem cells. The categories of research required review included HPS derived by any means. So some models for oversight. You could have no escrow, with the federal requirements in your institution monitored by contracts compliance departments, the Animal Welfare Committee, the IRB, the Biosafety. Or you could have a SCRO with a broader role, or an escrow with a very distinct limited role. Stanford has a joint SCRO IRB review process, and there are other kinds of models. So there are very many ways to do this. And I'm going to pass you on to Amy to tell you a little bit about Rockefeller University.
Thanks. A little bit of background. I've been involved in supporting the stem cell researchers at Rockefeller University since about 2001. We have a researcher who was interested very early on in developing human embryonic stem cells and looking at molecular aspects of pluripotency. So was working with embryos and with the goal of looking at the inner cell mass cells and then making the cell lines long before the NAS issued guidelines. The university also was involved in looking at how this type of research might be handled. We put together a interinstitutional meeting in October 2002, and it was scheduled to coincide with the issuance of the white paper that talked about the scientific molecular basis for determining something was pluripotent. So you had a scientific group looking at defining pluripotency, and then we had this meeting to look at the administrative aspects and compliance aspects that this new field of research was representing. So we had tech transfer people, legal people, development people, research compliance people coming together to talk about how we might want to look at this research. So that was 3 years before the NAS guidelines were issued. Because of the research that was being contemplated at Rockefeller, our president put together a bioethics group with a specific focus at looking at the issues that the proposed generation of embryonic stem cells represented. And there was focus looking at the provenance of the embryos, what types of consent, what were our concerns about the materials that were going to be used. And then there was also a big focus on transplantation into animals. And the role of that committee, I was a member of that committee, was to provide recommendations to the president. And we were to look at any existing laws, any existing guidelines, but we were an advisory body and policy was set by the president. The president allowed that this research was going to be conducted at our institution, that it was allowable, and probably the most prescriptive policy that came out was one about the transplantation of human embryonic cells into mouse blastocysts. So we had embryonic chimeric work going on in 2003, 2004. And the recommendation from the ethics group was that that should move forward cautiously. That the research should start where the lower amounts of cells being transplanted, a shorter gestation period, and then an evaluation of how much incorporation of the cells, and then have that looked at, have the researcher report back to the ethics group and then stage it so that eventually he was able to show that the amount of incorporation was small. We still know that from any of these studies that have been done, and that it didn't increase over time. So you ended up allowing for these animals to be brought to birth. So the Academy's guidelines came out in 2005. They recommended a formal oversight committee, and they gave some guidance on who might sit on that committee. So the NAS guidelines were informative for the university on how it might move forward. And they happened to coincide with a gift from the Star Foundation to the tri-institutions, which are the Rockefeller University, Weill Cornell Medical College, and Memorial Sloan Kettering Cancer Center. And that led to the formation of the Tri-Institutional Stem Cell Initiative, which still exists. The condition of the money for research from the Star Foundation was that it be collaborative across at least 2 of our 3 institutions. So the executive leadership of our 3 institutions endorsed the recommendation that we have a single escrow. It was going to be a brand new program we were setting up, bureaucracy we were setting up, oversight committee, and the concern was that if we had too many, if we had multiple escrows, that they might arrive at different decisions, and that would not be welcome. So we set up the Tri-Sci escrow. It is in the same format as it has been. It was established in the summer of 2006, had its first meeting in September of 2006, and it's 12 members, 6 internal, 2 scientists from each of the 3 institutions, and 6 external. We've been very fortunate to have long-serving members. We've had turnover in almost every single position, but members do tend to serve for a long time. We've been fortunate that way. Their actions and decisions are independent of the 3 institutions and cannot be overridden by any of the leadership of the 3 institutions. They look at all research involving or designed or expected to generate human embryonic stem cells. That's been from the onset and still is the case. After the 2008 amendments to the NAS guidelines and the advent of induced pluripotent stem cells, they added a category of work involving IPS that they would look at, so transplantation of IPS into animals at any stage of development or maturity, use of them in vitro experiments designed or expected to yield gametes. And then it expanded again in response to something we'll talk about more later, to all research designed or expected to generate human gametes regardless of whether or not the starting materials are multipotent or pluripotent. Okay. So, I think some things that are interesting about our escrow is the attention that they have given to the NIH registry lines. The Stryfer article in 2008 led to the first critical review of the original Bush registry and the decision by our escrow that they would not allow use of several of those lines because they found that the provenance of them was problematic. And then with the current NIH registry lines, our escrow is very interested in the provenance, the presence of any third-party gamete donation and the provenance of that. So they do not accept NIH registry status on its own as sufficient provenance. They ask for review and assurance that if any third-party gametes were involved, that they gave consent to research as a minimum, and they prefer that they gave research specifically for stem cell research. So those are some ways in which our escrow is, I guess, very diligent. They have an evolving position on grandfathering. So the NIH registry issue is one example there. They don't necessarily grandfather in the NIH registry lines, and they are not necessarily going to grandfather the use of any materials under the allowance of the guidelines, that if they were obtained with appropriate legal consent prior to the issuance of the guidelines, that they can be used. So our escrow is looking at that more critically. And then we also had our escrow seek and be given authority for the the gamete generating research, I mentioned that, and they are now currently deciding whether or not they're going to seek to be given authority for review of any research involving embryos, regardless of whether or not the intent is to derive stem cell lines. It's an active committee. They've reviewed 164 protocols. No human subjects yet. We don't have anything that's moved to a clinical trial. But we have had 13 that involve generation of human embryonic stem cell lines, 5 of which use SCNT. And about 54 of these have involved administration of derivatives into animals, 6 IPS directly into animals, and 11 human embryonic stem cells into animals, but only 2 of those were embryonic chimeras. Okay, so that's a lot about the Tri-Institutional Stem Cell Initiative, Escro. The 2 topics that I'm hoping to get some feedback from y'all is has to do with the research involving human embryos and then the use of materials from repositories or historically collected materials. So in this case, the question, I have a PI, say you have a PI, that wants to study embryological development of human embryos in vitro. This is not because they want to end up with a stem cell line, it's because they're a developmental biologist and because the science and the techniques have gotten to a point where you actually have a scaffold and it looks like we can actually culture human embryos longer and keep them alive in vitro. So if you have a researcher at your institution who's going to do this research, what committees would be looking at it? Would your escrow be looking at it? Is there a role for your escrow? And what guidance would you provide to your escrow about how they would look at this issue? There is a lot of, I think that the President's Ethics Committee on the moral status of an embryo is probably one of the good documents to point people to. It gives good arguments about the different things to consider about research on human embryos. And the NAS guidelines, of course, reference not culturing past 14 days or the formation of the primitive streak. There are some other documents that refer to that 14-day limit, but I've never been able to find the real basis for that. There seems to be a consensus out there about the formation of a primitive streak, but why? Why are we stopping there? Has anybody else had either their IRB or their escrow B pose this question?
I think it's a very interesting question because who would look at it? Who would think to look at it? You know, our IRB has reviewed a repository. And they have rules for releasing, and I don't think anybody included that particular restriction. I think it was not thought of, and it's a very interesting question.
Well, in our case, we had a PI who has actually sought ethical review of this question, and at this point the escrow seems to be the most appropriate body, and our escrow is considering whether or not it is going to conduct that review. There is no other body at our institution that would tackle this particular question. And as the escrow is having conversations about the review of research involving human embryos, they're being very mindful of how this might butt up against the clinical world, particularly in IVF. Because people do work with embryos in that world and aimed at improving methods and techniques. So however the world and the escrow world moves forward on this issue, there's a need for respect for that boundary. We don't want to have the escrows be inappropriately injected into the clinical setting. And then The other topic that comes up because of the provenance issues that our escrow is currently tackling is it's the grandfathering issue. As I mentioned earlier, the NAS guidelines allow under the 3.6 that you can grandfather the use of older materials as long as they're consenting. They were obtained under conditions that were legal at the time. But the NAS guidelines also have very prescriptive terms about what should be in a consent form for materials that you're going to use to create an embryonic stem cell line. You heard Melinda reference the fact that the HPS, it's only a subset of those terms. The escrow has been looking at the use of fibroblasts from repositories for quite a while because they have been extensively used for creation of IPS lines. And as a result, there are a lot of well-established lines out there, IPS lines, that were made using older fibroblasts. And now you might have researchers that want to compare different modalities of pluripotency, and they want to compare to an existing IPS line. They want to create that new line using SC&T. How does your escrow feel about grandfathering the use of the repository lines for SC&T? Is there a higher standard? Are there different issues? Is the step of creating the embryo for research purposes to destroy the embryo mean that there's a different standard that we should apply and a different approach to grandfathering as a result? So has anybody tackled this question? Most people must have looked at this issue with regard to generation of IPS. Has anybody's escrow had a hesitancy in grandfathering the use of existing fibroblasts for use in IPS? So thank you. And I want to thank Coriel in general. They have been such good colleagues and citizens and have really been patient with me as I seek more and more information from them on behalf of our escrow. And the types of conversations that or communications and dialogue I've been having, that is not unique to the stem cell world. Scientific advances happen, you can't ever predict all of the possible uses that the materials might be desired for. And so I was talking with Suzanne yesterday about the dynamic of, do we go to broader and broader, more general consents and say, we're going to use this for anything, and if you're not comfortable with that, don't donate. And the tension between that and the only guidelines that we all look to, which are the NAS guidelines that are very prescriptive about what they want in the consent. So I'm going to hand over to Melinda now, who's going to talk about partners.
Okay, we really only have about 5 minutes here. I don't know how quickly the room is going to fill up. We probably have another half hour or more prepared and a lot of questions we have for you, but I think what I'll do is just briefly state some of the difference of a different kind of model that we have and the different kind of challenges we face. Partners is I'm just going to say some of it. There's so much I have here that I'm not going to go through. But we do not fund stem cell research. We do have a state law, and I'm not going to get into exactly what that is right now either. But I think what I want to, or a history, I'm not going to get into that. I think what I want to point out is that, that our institutional culture is such that the role of the IRB, they review all tissue research. And all biorepositories remain under IRB review. Most often, repositories require donor consent for broad sharing of tissue. We have a lot of language in our biorepository consent forms so that downstream research will be allowed. No commercialization. One thing we don't have in there that the guidelines suggest is the idea of being transplanted into another person. And I'm listening to so much that's going on here with regenerative medicine, and I'm thinking maybe that should be added if it's going to be a repository. that would be doing IPS. So I think that we may have to beef it up a little bit more than we have now. In our early days, we were very conscious not to exceptionalize stem cell research. We had a very narrow scope. We didn't review any of the use of differentiated cells, including neural progenitors. The focus in the early days was on derivation of HESCs, including SCNT, the provenance of the HESCs, and at the time, the full committee reviewed all animal research, including teratoma assay. So as we evolved, there was less institutional interest in deriving HESCs, and the focus was on iPSCs. Our scope was informed by the amendment, the 2010 amendments, and we really left the creation and most uses of non-embryonic pluripotent stem cells under IRB review. The fact that the IRB reviewed all tissue research, that amendments for not human subject research were accepted and looked at. Escro consult is there if needed. So we were very cognizant of mission creep, and as opposed to Amy's Escro, we've kept it narrow. We do review use of pluripotent stem cells for sensitive uses, such as creation of gametes and implantation into animals when it would contribute to the central nervous system or germline. I also have some scenarios to talk about. It doesn't look like the room is filling up for anybody yet. Do people want to stay and listen to some of it, or do you have other places to go? Yeah. Oh, we do have to. Oh, we have to what? Oh, we have it for half an hour. I could have. Oh my gosh! I thought we had until 1 o'clock. Oh, okay. Oh, should we go back and see if there's anything else I wanted to tell you about? A little bit about our history, but I don't think it's that necessary. We did pre-NAS guidelines. We're in Boston, and we're by the other Harvard teaching hospitals as well, and Children's Hospital, and there's many other institutions. And we talked about the idea of doing one escrow committee, and but then the compliance piece came in, and we felt that we moved away from that. All right, so here's a narrative. An investigator. This is the question I guess we already might have approached, because it was brought to me by someone. The idea that you wanted to use an existing anonymized. So I think we answered this one already. That was created under a waiver of consent by a collaborator, and he also wants to use a few other anonymized lines from the institution, from a consented repository. So basically, our IRB would look at it, and we would look at the downstream research, what he was doing, and most investigators don't always think ahead, and there's unintended consequences, and we give them a guidance to think ahead, but we don't hold their hand. So hopefully they won't be sorry that they went with the one that was with a waiver of consent, but sometimes they might be in the future. This is the mouse blackness question, which you've already talked about. We're gonna finish early here. We'll have some discussion. I have some good. So I wanted to talk a little bit about creating gametes. So there are several sources of tissue for deriving gametes. There are the pluripotent stem cells. There's discarded ovarian tissue. There's repositories for gynecological tissue, discarded tissue from pathology, reproductive tissue that has been banked for fertility preservation, which if it's experimental, it would require IRB review, and discarded amniotic fluid. I think placental cell. I mean, there's a lot of different tissues that they're using to try to, with the idea of deriving gametes. So here's a scenario. An investigator submits an IRB protocol to use anonymized fallopian tube and ovarian tissue obtained from an OBGYN tissue bank. This is a true scenario. He said he was going to study differentiation potential. Now, some IRB reviewers have no clue what that means. So, not human subject research. They might not even think about it. So the IRB approved consent states that the tissue will be used for research on gynecological cancer and other problems, and that's what the consent form said. So what actions might your IRB take? I mean, is anybody here who sit on an IRB that they would think to give it a second look? I think this is one of the protocols that can just go through the tracks, not human slip through the cracks, not human subject research, and not be seen. So the investigators really have to be educated that the escrow wants to see these things. Oh, does the escrow want to see these things? Our escrow policy doesn't talk about fallopian tube or ovarian tissue protocol even. There's nothing in our policy that says it. There's nothing that would even alert the investigator to submit that to us. So when you're answering the question, there are a lot of issues to consider here. And one of them is that just for HPS cells that they aren't subject to any review beyond that necessary for any cell line, except that any experiments designed or expected to yield gametes should be subject to escrow committee review. The federal regulations, the NIH website on stem cell research, and this old OHRP guidance from 2002, there's no deviation at all from the current regulatory mechanism. At all. De-identified tissue, whether it's embryonic stem cells, germ cells, stem cells, it's all not human subject research. Waivers of consent are allowed for the use of identifiable human tissue for in vitro research. This is all still up there. Doesn't say that it's, not. State laws. Is it legal to create embryos for research purposes in your state? Is there intent to test viability? In Massachusetts, it's a criminal offense if you create an embryo for research by fertilization. So if you're testing out your gametes, you could be put in jail. And fined and put in jail. International collaborations. So would there be a reach-through to an international collaboration if they are going to, the collaborators in the UK, for instance, got the license to create the embryo over there? Or what is your engagement, what is your investigators and your institution's engagement in that research? This Hinxton report, this was a group of policymakers in Ephesus who got together to talk about the issue of creating gametes. And they came to this conclusion, which was very interesting. And our escrow, when we talked about it many years ago, before the reality of creating gametes was before us, it sometimes changes your thinking when the reality is really there, was that just because there are divergent moral views, you shouldn't limit the research. That's what they said. And they said specific consent would not be needed to use tissues to derive gametes that are intended for in vitro use only without the production of embryos. And I'm just going to skip down. There should be requirements for specific consent by tissue donors for use of the tissues to derive gametes that are intended for use in reproduction. So, it was a little surprising. The people on this panel were, people from Hopkins, people from some of the big institutions here as well as internationally. So I put this up, but there are other articles that say the opposite, that you have to respect autonomy, that people should have consent. This is something that many people would have the yuck factor for. So, this is the same one, right? Okay. Okay, so another investigator submits a protocol to the SCRO committee to derive sperm using anonymized human iPSCs obtained from your institutional iPSC bank with your IRB-approved consent form to derive gametes. Has anyone here seen any of this research to derive gametes? No. So again, I mean, our committee, a SCRO wouldn't see it. The Escro and the IRB would see it. And again, it's the downstream research that we would be considering. We'd look at the provenance, what the consent form said, and to us it's very simple. Our IRB looks at provenance for everything that comes in, and whether it's consistent with the use. So, it seems fairly straightforward when I look at this question to me. You look at the consent form, you look at what he wants to do. Okay. So, did anybody come here with any burning questions, or does anyone want to share something about their institution? Kathleen.
You mentioned that this whole issue of mission agreement comes up, in a lot of discussions. In your institutions, how are you keeping that mission creep from expanding into? Is it primarily because the jurisdiction of a lot of these issues remain with the IRB and don't become part of it?
I think the IRB. The director of the Escro is also the director of the Human Research Office. So we sit and we discuss these things together, and sometimes some of us think maybe it's time to do what your institution's doing and ask for a broader scope. But so far we've been resisting it. The idea was, why we resisted a lot of things. That we don't want to be the embryo police, but who is? Amy brought up a point. We're there to protect human subjects. We're not there to think about any of the moral issues. But so, yeah.
Just one more question. What, in a general sense, what is your faculty's appreciation or lack thereof of the existence of Escro?
I think that right now our Escro is the focus is so much on IPSCs. Right now we are really providing a lot of guidance on whether their federally funded research can use certain lines, giving guidance on we have a guidance on use of pluripotent stem cells, so we're letting them know when it has to come to us. I think it's not something they think about. We don't, we're not extremely active on any burning.
Look to you as a piece of an advisory.
Right, yeah. So that's that. So I want to know whether you see, let's say I see. Is anyone? Are you work for an IRB or an Escro?
What?
Are you involved with an IRB or an Escro? Okay. So so I want to know from people here and
I do clinical research also.
Okay. All right. So I want to know, do you see the roles of your committees expanding, taking on more, or Kim?
Self-research unless it's in I, as a director of this, question the relevance because not much comes to me. And we've talked about it at our committee, whether we just don't have relevance. And the scientists actually feel that we do because we've been a group, like you were saying, that have been together for quite a while and seen sort of the evolution of the science. That eventually, with the other factors, it's important to have a group that has been together a long time, or that may happen.
Thank you. Are either of you from Texas, by any chance? Oh, you're from Texas. Do you have a sense of what's happening here as far as, because I see that it's very active. There's a lot of research happening. Through existing, like on the NIH registry. Okay. So then it can just go to the compliance office. It doesn't need a whole committee to look at what's happening. Okay. Do you have any questions? Thank you for joining us. Yeah, thank you very much.